Bacterial transformation is the process in which we introduce DNA, typically in the form of plasmids, into bacteria. And for us, we're going to use some sort of cloning strain of E. coli. Now, later, we're going to learn how to do a yeast transformation, very similar to a bacterial transformation with some minor nuances. But generally, this is going to be something that we do fairly regularly. So here's my amazing drawing of E. coli. This is some sort of cloning strain of E. coli, and we'll learn the names of some of these a bit later. And on the right, we have plasmids. Now, plasmids are small, circular, independently replicating pieces of DNA, and they have very special properties. And for this example, the most important feature is the presence of an antibiotic resistance gene, or what we call a marker. Now, there are many different types of markers. Typically for bacteria, they're antibiotic resistance genes, but you can have nutritional markers. So, for example, genes that are involved in amino acid synthesis or nucleic acid synthesis. And regardless of which type of marker that we use, all markers do the same thing. It helps us ensure that the E. coli or the cell in question actually receive the plasmid that we are adding. And we'll see that in a little bit as to how that actually works. So the question then becomes, how do we actually introduce the plasmid into E. coli? And we can't just stick it in. It doesn't quite work like that. So it turns out that we need to make our cells what we call competent. And the ability called competence is the ability to take up DNA. And there are many naturally competent bacteria, but E. coli is not naturally competent, so we make it artificially competent in the lab. And this is accomplished by coating the E. coli cells with positive ions. And the reason that this works is because DNA is largely negatively charged. And so when we mix our plasmids with our competent cells, the negatively charged DNA ends up sticking to the positively charged cells. Now, this in and of itself doesn't bring the DNA into the cell, so now we need to force it in there. And we do that typically through a heat shock. We can use other methods like an electric shock, but a heat shock generally shocks the cells and it causes the DNA to flip to the inside, and then allowing the cell then to take up that DNA. And so this is the process that we're going to see. We'll kind of remember these steps, and we'll talk about each of these steps as we see it in action. So competent cells are very sensitive, so we keep them in our deep freezer. The strain that we're going to use for this example is called DH5 alpha, and whenever you handle competent cells, always keep them on ice as much as you can throughout the whole process. Now, before you get started, there are a couple of things that you should get ready because these take time. One is to set your water bath. So simply turn on the water bath, make sure that there's water in it, of course. Hold the set button and hit the increase or decrease button until you reach 42. It will take a little time for the temperature to get there. Make sure that the lid is closed the whole time while you're waiting for the temperature to equilibrate. And the other thing you want to get ready is the Soc medium. This is typically in the freezer. It is frozen, so it will take a little while to thaw. So once again, just pull it out and let that sit on the bench. So this is Jaden. He's been working on a particularly tough cloning assignment. So we're hoping that we can get a successful transformation, which means that the plasmid has been formed and the gene has been cloned. He allowed the competent cells to thaw on ice. Takes about 15 minutes or so. Remember, gotta be gentle. And he takes his cloning reaction, this is a Gibson Assembly, I believe, and pipetting it and mixing it with the cells. Now notice after he pipettes, you do just a gentle mix, right? You don't want to go a crazy flick mix. Just kind of tap the sides of the tube. Definitely no vortexing. And just a bip, 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 bip. Something like that. And then let it sit on ice. 30 minutes, longer the better. After incubation comes the heat shock. Bring your cells to the water bath. There's generally going to be a rack like this that floats. If this one isn't present, there are others available. Have your timer ready. Place your cells in the rack and then 45 seconds. So your heat shock can go anywhere from 30 to I've seen as high as 2 minutes. Generally 45 seconds works for most of the applications that we use in our lab. So 45 seconds is the one that Jaden is using here. After the shock, it's important to plunge the cells straight into ice. Allow the cells a little time to recover. They have been quite stressed during this time, so let it sit on ice for about 2 minutes. After the heat shock stage, we hope that the cells have taken up the plasmid DNA. However, they're still quite weak. So this next step, which is called the outgrowth step, allows the cells to grow for 1 generation. This allows them to fix their cell walls, copy their DNA one time, and just be ready and more robust to actually resist the antibiotic. To do this, Jaden will add some Soc Medium that he let thaw. The amount that we add can vary. I've seen as low as 150 microliters up to as high as 1 mil. We tend to use 450 microliters, which is a little bit on the high end, but again, it works for us, so that's what we're going to continue to do for at least for a little while. So gently pipette in the Soc medium, and then we're going to incubate it. And we want to have some aeration or some shaking while it's incubating. So to accomplish this, grab an Erlenmeyer flask and take it to the incubator. Make sure you turn the incubator on. It needs to be set to 37 Celsius. This is a kind of a weird incubator. You actually have to turn the on button and then use the up and down arrows to set the temperature. Place your flask. It's a lot harder than Jaden makes us look. Into the holder. Close it and then give it some shaking. And so we're just going to simply let this shake for about an hour. And then that'll allow the cells some time to recover. It's going to start slow, but it'll get there. The last step is to plate your cells onto agar that contain the appropriate antibiotic, the one that is on the plasmid. To do that, simply plate a certain amount of cells onto the agar. The amount is going to vary, and it's always going to be dependent on what the expected transformation efficiency. And later I'll show you some pictures of what it looks like when we have an inappropriate amount. In this case, Jaden is using 50 microliters. Right onto the middle of the plate. And then he's got a spreader soaking in ethanol. The ethanol is going to sterilize the spreader. Simply tap it off and then pass it through the fire, making sure that the ethanol burns off. Usually I pass it twice. If on the second pass you still see a little bit of that blue flame, then I'll pass it a third time. Cool the spreader by touching it on the 4 edges around the drop. Don't place it in the drop, otherwise you'll sizzle the drop, right? You just want to make sure that the spreader is cool, and then spread in a circular motion. Jaden is going the same way. I like to spread in a clockwise direction and turn the plate in a counterclockwise direction, and slightly larger circles than what Jaden is doing here, but at the end of the day, this should work just fine. Keep on spreading until you feel a little bit of resistance as the liquid starts to get soaked up in there. It'll take about 30 seconds or so. Once that's done, put the spreader away, close the lid, and we're good. So in our hands, generally we plate a small amount, somewhere between 50 to 100 microliters first, and then we plate the rest of the transformation mixture. Now, the problem with that is that the amount is quite large, so we centrifuge it real quickly, 1 minute at maximum speed, and then you just dump out the remaining liquid. Now, you will always have a little bit of liquid left over, and so we can actually use that to resuspend the cells. So Jaden dumped out the liquid, and then now once he gets his pipette box open, the tip box, simply use the remaining liquid in there, gently pipette up and down. Usually the pipette here is set to 100 microliters or so, because that's about the amount that will be left over, and just gently pipette up and down, making sure that the small cell pellet, and it's going to be a small cell pellet, gets fully resuspended. And then once it's fully resuspended, plate all of that mixture onto the cells, and then spread as you did before. These plates are now ready for incubation. Simply walk them over to the incubator, set at 37 degrees Celsius. Place your plates in there. Make sure that they are lid down, always lid down because of the condensation. And we'll see these tomorrow, crossing our fingers. And since Jaden couldn't be here the next day, I had to give him the news of failed transformation. So no colonies present. That's no good. I did take some better pictures, and so let's see what they look like. So unfortunately, no colonies on here, so this was a failed cloning attempt. Like I said, this has been a particularly tough one for, I'm not even really sure why, but Jaden will go back to the drawing board and we'll try it again. So let's look at what some successful transformations will look like. So just to show that Jaden can indeed clone things, this is one of a successful transformation. And those little dots, in case you didn't know what they looked like before, those are colonies. They're mounds of bacteria, and since each of these essentially rose up from one single cell, each of these colonies are what we call clones. They are theoretically unique individuals in this population. And theoretically, each of these individuals can have a different construct or just a different way that things were cloned in there. So generally, when we screen things, we'll pick multiple clones to screen for the presence of our properly formed plasmid or properly cloned gene. The amount that we screen will often vary on the types of cloning that we've done. The much more efficient, higher success rate cloning, we need to screen less because we expect most of these to be the correct construct. The ones that are a bit more challenging, sometimes we'll have to screen more just to make sure that we find it. Now, the reason that we plate different amounts of bacteria during our transformation is because the amount of colonies that we see is going to be dependent on the type of cloning that we've done, the type of transformation, the type of Copton cells, etc., etc., etc. So on the left here, we have that 50 microliter one that Jaden plated. This one turned out to be a very nice amount because we can see a decent amount of clones. These clones are very well separated, whereas the resuspended version, or the one that he spun down and then plated the rest of, you can see that just way too much. So you can't comfortably pick an isolated clone. Now, here's an example of a cloning attempt that was a little bit too efficient. You'll notice that even on the 50 microliter plate, there are way too many colonies. So in this case, we probably would have to plate it less or even do a dilution prior to plating. Now, it's better to have too many colonies than no colonies, but still at the same time, sometimes it's one of these things where you just have to kind of know the cells that you're using and the cloning attempts that you're doing or the cloning type of strategy that you're using in order to find the right amount to plate. And last one here, this was a gene that Ashley and Tiffany have cloned. Sometimes what we do is we actually pre-pick our colonies prior to picking them. So they've gone ahead and circled the colonies that they wanted to pick. And this way, when it comes down to actually picking the clones that you want, they've already kind of indicated the ones that they would like to get. And the benefit of doing this is that sometimes if you end up losing your grid plate or the clones that you have, you can simply incubate this plate longer, which is what they did here. And then the little scrape that you made on the plate will grow back into larger colonies. So you can recover the clones just in case you lose them. It's an optional thing. It's a really good practice to do, but certainly not something that we do all the time. And there you have it. That's transformation. We'll show you how to screen for clones and determine whether we have the correct clones very soon.